it is not like other technologies and conventional field where you have very much standardized workflows and technologies. Unlike that, in proteomics, there's so much innovations going on, lot of new technologies, new methods, new softwares being developed, and also how people are innovatively utilizing that to address some very challenging biological questions. So therefore, the field of proteomics is very rapidly evolving, and to keep up the pace of learning, sometimes it's a good idea to also attend some conferences and workshops, which gives you a lot of information in a very short time. So I recommend you to attend one of the Human Proteome Organization conference, which will be, I think, happening in October, and that is this year being online, virtual Reconnect conference. I think that opens up more opportunity for more people to get engaged and get the knowledge. Otherwise, we used to travel to different countries to attend this meeting, and only some people who are there can gain that knowledge. But online platforms have opened up more possibilities now that anyone actually can attend from anywhere and gain the knowledge. So I think that will be probably one of the informative meeting. If you're interested, we'll share you some more detail. But coming to today's class, let's start about the sample prep and the gel-based proteomics, and then moving on to the some of the activities which we thought for your project workflows and discussions. So after giving you some overview recap, then I'll try to finish the sample prep which we were discussing in the last class, and then we will take the what is the workflow going to be for your project and presentations. We are going to give you some data for analysis and then have some discussion. So just to recap, I think it has been now almost 11 lectures by now. We are walking you through different type of proteomics based workflows, and primarily focusing about mass spectrometry as one of the technique. However, the data being generated, whether you generate data from microarrays or mass spectrometer, or from the 2D gel, dye gel, any of those, finally leads you to a list of possible protein, which you have to make sense of that data. And for doing so, there are many software which we have taught you in the process, which is all going to be very much utilized, irrespective of which workflow we you will be exposed for different technologies. You have to use some of these resources. But also, I think probably you'll appreciate that many of the software and the databases which we are exposing you, those are all pretty much online, open access available. So anyone can access anytime from anywhere. Otherwise, earlier we used to have only commercial software for doing some of these analysis. That was very cumbersome, as you can understand, and that was not straightforward for everybody to access those stuff. So only in the class we used to show those softwares, and people can be given access to doing those analysis. Now it is available from anywhere for that matter. So we have talked about discovery workflow, quantitative proteomics. We talked about pathways and network analysis. We talked about targeted proteomics, and then I'm touching upon the sample prep. Gel-based, I will briefly talk now, but it will be essentially in the next class. But we thought to start discussion about the projects even slightly ahead, so that you are getting exposed for these things ahead of time, preempted ahead of time, and you start thinking about which topic and how to take that forward, because post midsem you will have less time to start preparing for the presentations, and therefore we want to be fair to everybody, including the first and second group of students who will be presenting on those days, so that you also have enough time to get ready beforehand. This is where we are right now, and what we are talking in the previous class was essentially how to prepare good protein extract, good protein sample. And as you would have appreciated that there were a variety of sample which we talked about, and every sample type required different type of workflow, different type of thought process, different type of contaminants are there, different type of interfering substances are there. We have to know about them and we have to remove those. After doing your proper lysis and precipitation, reconstituting the pellet in the right buffer, then you are curious like how much protein quantity you got, and that's what you did the protein quantification earlier. Now finally you want to look at how good your gel shows the protein quality, the protein extract which you have extracted on the gel. Can you see like does that provide you good quality? For doing that, you can separate the proteins in one dimension gel known as sodium dodecyl sulfate polyacrylamide electrophoresis SDS page. I'm sure most of you have already done in this particular class who are attending this course right now, but some of you who have not attended, it is one of the electrophoretic method, which is going to separate the proteins based on their molecular weight. And you are going to separate only one dimension based on the molecular weight. You want to denature all the proteins and then separate the proteins in the electric field based on the molecular weight. You add the detergent SDS, which makes it negatively charged, and therefore in the electric field it can move. So now I will again talk about the principle of this and the each reagent which is required to make SDS pH gel, and what is the role of APS, timid, and stacking and resolving gels. All that in the subsequent class when I talk to you more in detail about gel-based proteomics. But right now, in the context of checking the sample quality, let me show you the gel apparatus and the setup. This is what we are talking. You are preparing to check the quality of your given sample, and if you are to come to the lab and do this experiment, you are going to first cast a gel. And this gel is, you are first taking the glass plates, a thick and a thin one. You are placing them inside the assembly, and you make sure that the level is flat, because otherwise, if there's any leakage, it will be a problem. And then you put them on that floor gasket which you saw, so that it is tightly placed on in that gasket. And some of these units are very user friendly, when you will not have leakage of the liquid. So you are preparing this particular recipe with the with pH 6.8, and you have the timid APS. You're adding all of these to make this cocktail, which then you are going to add in this particular assembly, which is having a thick glass plate and a thin glass plate, and there was some small notch there in between, so that the liquid can stay there. And you want to make sure these kind of bubbles are not coming, otherwise they are going to create a problem in that well. So first you put the stacking gel, and then after that, so you have done the resolving gel, and now you're putting the stacking gel on top of that. And then you are going to place the comb, which is going to create the wells over there. So these comb is essentially after polymerization of the gels, what you are adding is the solution form. And then after addition of timid and APS, the polymerization happens. Then since you have added the comb, it is going to create these kind of wells in the gel. So once you remove this comb, then you will see like that empty space is there where you can load your protein of interest. So after loading this particular protein which you are now adding there, to visualize that how the run is going, you're also adding the dye which is bromophenol blue, so that while the electric current will be passed, you can also monitor the dye how it is moving in the electric field. So in a typical gel, you will have almost 10 sample which you can load. And as you can see, like with the pipette, maybe 5 microliter or 8 microliter maximum you can load in this particular wells. And after you have done the loading, then you can add the buffer, which will be separating these proteins in the tank. And you set up a high volt, I think 90 or 100 volt. It will take at least 2 hours or so time to separate this particular gel. So at the end of this process, which will be, this particular gel will be done, what will happen that, you have run the gel, which nothing is visible other than seeing a different, like, that gel is running, nothing you can see on the gel. So then you take this transparent gel and keep in a dish like this, where you can add the staining solution. And then after adding the staining solution, you keep it overnight, so that with the Kumasi stain or whatever stain you have used, the protein is going to bind and show the dark band pattern, but that will not be visible after staining. You have to de-stain the gel so that the gel is removed, and whatever bound protein is there, only that will be visible. So then at the end of the whole exercise, you are able to see this kind of gel pattern, which is the typical SDS pH gel. The first lane, usually you keep a known standard markers, like these are the protein for which we know exactly how much is the molecular weight, like 70, like 100 kilodalton, 75, 35, like that. So you will know exactly, and so that you can also try to align that how good your proteins from the high molecular weight to the low molecular weight are able to separate on the SDS pH gel. Now, if you have prepared many sample, what you can infer from this gel, that this sample looks good, even this sample looks good, this is also good, this may be okay, but not so much. This one and this one may be okay, but this one, this and this, these three are good sample. What we have also learned that this sample and this sample is not good, this sample is also not good. and few samples are on the borderline. So there could be two possibilities and two problems. One is your extraction method did not work out so well for these samples, or you might be comparing right now different type of methods, like TC acetone precipitation versus acetone precipitation versus trizol. You might be comparing different methods as well and seeing which method is giving me the better yield of my protein and good type of quality of the bands on the gel. What we are looking at a uniform pattern of the protein from the low molecular weight to high molecular weight. Throughout we see the sharp bands coming on the gel. That's what you want to see. You don't want to see something like this when it looks like a smear. You don't have many bands here and looks like either protein is degraded or very less quantified, and we don't have sufficient amount of protein on this gel. This is also decent gel, which shows that very uniformly we have sharp bands available. So any kind of sample prep method will bring its own type of bias, its own type of innovation, sometimes good or bad. But finally, you have to check those things on the SDS page gel to make sure that the quality of gel reflects what sample quality is there. And sometimes you need to then re-quantify some of these samples again, rerun this sample again, and make sure that you are happy and confident about which samples you're going to take forward, which extraction method you're going to take forward. So this was the QC check or quality control check on SDS page. As I also referred in the previous class, the same sample could also be run on an agarose gel, like the same protein what you ran here to separate the protein here, but on the agarose gel it will be you are going to separate the nucleic acids. And if there is any contamination coming in this sample, for example, this sample might be having too much nucleic acid and very less protein. So when you run this on the agarose gel and stain with the ethidium bromide, you might be able to see there's a lot of things get clogged in the well, whereas in the other samples maybe it is very clean and there is not much a contamination of nucleic acid. So it's also good to cross check those. And of course, you're looking at the protein quantification as well. All these steps are very much important before you proceed for the next step, right? And next step will be like this. So now refresh back your workflow, which we talked in the mass spec based proteomics. We have talked by now different ways of cell lysis and sample preparation, doing quality control check on the gel. So now you can see this kind of gel. And then we have been talking about this entire workflow. I hope now you can reconnect and you can recap and you can think about the entire workflow of what we have been discussing and how things start and how things finish. We have put a lot more time and emphasis on this part about data acquisition, data analysis part. And then that series of steps goes on before that. We have showed you different type of instrumentation directly from the lab. So this is part where we are right now, like from the protein extracts. Now we are going to digest the protein so that the peptides will be prepared, and those peptides are going to be injected in our mass spectrometer for further separation and subsequent analysis. So question comes like, why you need to digest these proteins to make peptides? Because ideally you have already extracted the protein. You can simply put them in the with the liquid chromatography and continue them with the mass spectrometer. So ideally I would like to ask more of this question what I have in my slide, but let me now make it more just broad overview for you that since we are talking about different type of mass analyzers, and as you have learned that different mass analyzers like quadrupole versus time of flights are very different in terms of what kind of masses they can separate and they can resolve. So many of the mass analyzers can only take the limited mass range. Only TOFs are quite exceptional in that way. So if you can digest the complex protein of very high mass and then try to make the peptides which will be having the less mass range, then any type of mass analyzer, any type of mass spectrometer can analyze your proteins of interest. So that's where you are trying to bring everything to make it shorter so that it can come within the analysis range of the mass spectrometer. So that's why we are limited with the proteolytic digestion. And enzyme digestion can be done using different type of enzymes, different proteases can be used. Trypsin is one of the very efficient and very highly used enzyme which we all use in our labs, which ideally cleaves any polypeptide chain with the lysine and arginine amino acid residues. Only if there are some proteins which contain the proline residue, then there will be a problem, and then you will have to look into the, if just before lysine and arginine, if there are proline, then that may not be sufficient to digest your protein with the trypsin. and therefore you have to look into these sequences as well of your proteins of interest. Finally, a typical digestion, what people in my lab do, at least 15 to 20 microgram of protein is taken, and then that being digested for subsequent experiment. But even one could go as low as even 1 microgram as well. And you have to maintain the enzyme and substrate ratio. So enzyme trypsin versus your substrate, which will be the protein coming either from the gel or from the solution form, that you need to keep at least 1:30 ratio. So these kind of things you have to keep in mind. And sometimes there are other alternative enzymes like Lysine is there, and endopeptidases are there. Some of these type of enzymes can also be tried out if your proteins have some anomalies, especially with lot of prolines rich. Therefore, you can try even the modified trypsin, or you can use the lysine and other enzymes as well. So ideally, what is happening that your complex protein, wherever there are lysine arginine residues are there, trypsin is going to cleave that and going to make multiple of these type of sequences, and these are the peptides which are going to be subsequently injected in our mass spectrometer, and these are the ones which are going to get ionized in the electrospray ionization, followed by LC and MSMS separation. So now in between this process, there is one more crucial step involved. For example, you have prepared your protein, you have done the QC check of the protein, but then now you want to inject that protein to the peptide forms in your mass spec. So you have to do either in gel digestion or in solution digestion. In gel digestion, when I say it means you have separated your proteins of interest, let's say on SDS page gel. And many times, many students project that simply revolves around looking at one or two specific proteins. Like you are purifying a protein of interest, or you are keen into looking at a specific protein molecular weight. So let's say you are excising this band of interest, which is what you want to know which protein is that. Or alternative way could be that you are interested into the entire complex proteome, and you chop down the entire gel into 10 pieces, for example, and take each of those gel pieces and do the in-gel digestion of each one of those, and put together data, and then now you say this is my yeast proteome coming from this particular gel lane. Or you are interested only in a specific band of interest, or you are interested only in a specific spot of interest, and these things I'll talk in the 2D gel in the next class. So in either way, what you have is in a gel piece of SDS page, acrylamide gel, you have a small amount of peptides present from the proteins of interest, right? And these proteins you are going to digest to get the peptides out from that, and then those peptides will be subsequently analyzed in any type of mass spectrometer, whether it's multi-TOF or ESI Q-TOF or ESI Orbitrap. All of that can be utilized to analyze this sample type. So in gel digestion, again, you need not to remember all of these concentration and the reagent detail, but I'm just kind of giving you some idea for the if you were to do this thing in the lab. First is the gel piece has to be de-stained. So you add ammonium bicarbonate to do that process. You want to make sure that while you are processing this gel piece, it should not get dried. So the gel should be completely all the time keep in the wet condition with the acetonitrile. Incubate that for 5 minutes at 37 degrees. Then do reduction step when you can use dithiotretol, which is going to break open all the disulfide bonds. So because you want to finally cleave all these bonds to get the protein digested in the peptide forms, an enzyme has to work. So now after this reduction step, then you can add the alkylation step, which is the idostamide, which is going to make sure that the sulfhydryl groups are blocked, and it is not going to reoxidize and reform those bonds of disulfide. And then you are going to make sure that after each step you are dehydrating your gel pieces with the acetonitrile. And then you are adding the enzyme trypsin, which you have to follow certain rules. You should know how much amount of protein you are going to digest, how much enzyme trypsin you should digest, what is the enzyme substrate ratio being maintained. And then depending on the type of trypsin which you have from different vendors, you have to then incubate that for either overnight or for 4 hours or for 1 hour. Just now we have used one of the enzyme trypsin, which is a very rapid digest trypsin, which can be used for even 1 to 2 hours of digestion sufficient. But most of the trypsin which comes from other commercial makes, those are typically used overnight. That gives sufficient time for these enzymes to work on the your gel piece and on the protein to make them into the peptide forms. So that is one of the ways of doing your gel separation. Second alternate way of doing this could be in solution digestion, and let's see this in the
For the mass spectrometric analysis, in order to identify the proteins in a sample, peptides are required. For getting the peptides, the protein sample should be first digested. Protein digestion can be performed in a gel-free or a gel-based manner. We will show you how to perform the in-solution digestion, that is gel-free procedure. So now, after quantification, suitable volume of the sample is taken for digestion. Before doing the actual trypsin digestion, we need to process through the two steps, that is reduction and alkylation of proteins. For reduction, here we are using TCEP, that is tris-carboxyethyl phosphine. Appropriate volume of TCEP is added. The sample is properly vortexed and spun. After addition of TCEP, that is the reducing agent, it should be incubated at 37 degrees for 1 hour. So after getting incubated at 37 degrees, the samples would be reduced. So after incubation at 37 degrees, reduction is happening. That means all the disulfide bonds are broken within the proteins. So now it's time to alkylate the sample. For alkylation, iodoacetamide is used. During the alkylation procedure, alkylation will block the thiol groups so that the disulfide should not be reformed. After this, the sample is properly vortexed and spun. To perform the efficient alkylation, the sample should be kept in dark for 10 to 15 minutes. So after completing the steps of reduction and alkylation, the next step is addition of trypsin for the digestion. So before that, the step is dilution, which means we first need to dilute the molarity of urea that was present in the initial sample. So the diluent is used for diluting the sample. Appropriate volume of diluent is added to the sample in order to dilute it. Before addition of trypsin, make sure that the pH is 8 so that it can work properly. Now the next step is the addition of trypsin that could be added in the ratio of 1:30 to 1:50, which means that one part of the trypsin can be sufficient for 30 parts of protein. The sample should be vortexed properly and spun. And finally, for optimum protein digestion, the samples need to be incubated at 37 degrees for around 16 to 17 hours. After 16 to 17 hours of incubation, the protein sample is completely digested, and then the sample is dried in order to get the peptide sample. These dried peptides can be taken further for mass spectrometry. For mass spectrometry, the peptide sample should be properly cleaned. To make sure that the sample is clean of various salts or other contaminants, the sample can be performed through the cleanup steps. For these steps, we use C18 columns. So these columns can be made using the C18 material within the tips. And then the sample is passed several times through the column. This column works on the principle of reverse phase chromatography. During several steps of passing the sample to the column, the peptides would be bound to the column, and then finally the peptide would be eluted. After the peptides are bound to the C18 column, they can be further eluted using different gradients of acetonitrile. And then the elutes is again vacuum dried, and the sample can be taken for mass spectrometry.
All right. Well, thanks, Sushmita and Meher, for showing you the steps. I hope all of you got the visual feel of how to do these experiments in the lab, which is not very difficult. You just have to add, the reductant and alkylating agent and enzyme, and ensuring that in the process the everything is maintained with the right temperature and right type of timing for incubation. And if you do that all well, then I think you can get good digested peptides from the protein. So that's all for the sample prep activities. Let's move on quickly now to discuss a little bit about your project activity than what we have planned for today. So we are going to show you some steps, and you are going to be also doing some steps. First thing is, broadly, we are going to give you two type of projects. The first type of project will be for most of the students who are with the bio background or towards more PhD and this kind of program. And second type of project will be more for the some students who are more from computational background and the UG then MTech students. So this type of project which we are talking right now, we are going to shortly give a demo file to all of you anyway, so everybody should do that part. And what we are going to do, the things has to be kept on very much time because we will be having so many steps involved. So we are giving you a file which in 10 minutes time you have to do the MetaboAnalyst from that file. And each one of you will do on your own system and you will see what output you get. Today there is no quiz, right? But we do want you to do this analysis because we are going to walk you through how you are going to do your project. So after 10 minutes, we will talk to you about your data output, what you got, what you should be getting from that data. So if any queries, we can resolve that. Then we will give you same data you already have by now. You take 10 minutes time to do Reactome or string analysis from the same data. And then we will discuss about what output you got and what is expected from that output. Further to that, from your MetaboAnalyst and the Reactome, you may have identified few targets or pathways of interest, and you want to learn a little bit more about these proteins. Now, can you take the Skyline or targeted workflows or NextProt, Human Protein Atlas, many things which we have talked for protein better understanding. Now you can go through some of those software tools and then tell us that, okay, what is your understanding that how well you can take these leads forward with these softwares. So again, we are going to give you some time for you to do these things yourself, and then we will talk about what is the output. So this will be done by every student in the class. Second type of project will be more computational and programming oriented, which we are only going to give you some overview that what is expected from this kind of project. We are not going to give you a file for you to analyze right now, just matter of giving you some exposure of it. At the end of this exercise today, before we close the class, we are going to give you a Google form, a very short form, which will give you only two options. Are you interested in project type 1 or you are interested in project type 2? We have very heterogeneous students here in this class, and we want to know that how many people would like to take biology type of project or looking at the clinical aspect of the disease finally from the data, and how many people would like to do computational projects more in silico analysis, transcriptomic analysis, which can be so again, once we know these type of number of people who will be engaged in these projects, then accordingly we will come up with the topics for giving those many students. For example, if there are 30 students take this type of projects and 10-15 students take this type of projects, we have to then come up with my team has to come up with the ideas of those projects which will be assigned. Or if all students are going for only one type of project, then we have to also come up with that plan. So just to give you a little bit heads up why we are doing this exercise, essentially, as you are aware that we are going to ask you to make a presentation based on your project work. And at the end of that presentation, everybody in the class is supposed to understand that topic and do some write-up on that. So we are going to assign you one unique topic, right? A unique topic means everybody will get a separate type of data set, and you have to do that project yourself. We are going to walk you through today, and in subsequent class also we'll have more discussion. So you are getting enough time to prepare. Based on these, then this weekend we are going to send you a form, Google form, with the list of topics. So today we will know that how many people want type 1 project or how many want type 2 project. And then in the subsequent days, we will assign you multiple topics. We will give you a lot of topics and options. And then you will select two topics. One is the first option and one is the second preference. Based on that, we would like to assign you the topic, which now you will be given a data set. And you are going to analyze that data set from the scratch. And then you will do some literature review to understand that what that disease and that protein means, because we are going to give you a problem statement. For example, the role of a given drug to treat a colorectal cancer cell line. That is the problem statement. Now, after doing the proteome analysis, you want to know what was the effect of the drug and how that has, controlled or inhibited or, has no effect on any pathways linked to colorectal cancer. Or for that matter, different grades of a given tumor. how disease is progressing and what kind of pathways is being changed. So some of these things you have to then come up independently from that problem statement. And then when your turn will come in the class, which will be essentially post midsem, then you will have to make a 15-minute presentation followed by 5 minutes question and answer session, which will give you quite substantial marks, 20 marks. So therefore, this is very important exercise for you to do that. And based on this, you're also going to have some write-up, which will also give you marks. So that is almost 18 marks, which will be coming from 2 marks of each class. Now, every class, there will be 4 presenters. So each one of you will present for 15 minutes time. And then 5 minutes question answer. So 80 minutes will be like that. Now, after, let's say, today's class, 4 presenters have made presentation. In the next class, we will first 10 minutes, they will give you a Google form, where based on each of the 4 presentations, there will be 1 or 2 short questions which will be asked, so that you will we would like to know your understanding from those presentations, because we do want everybody to attend all the presentations and evaluate everybody. We are also going to give each one of you the marks scheme, that you have to also give marks to your fellow students, which will also consider in the our final evaluation. So it will be more like peer review process. We want to see how you as a student evaluate your peer group of the students who are of the same age and same type of level. So this will be a fun exercise, and I'm sure all of this part will really comprehend what all you are learning in the course so far. Everything you will be start comprehending independently, and at the end of the exercise, you are doing a project which will be a very unique project. You are going to make a presentation based on that project, and some people are going to write some statement and understanding what they have learned from that presentation. That's all. So far today's lecture and the previous lecture, what I try to emphasize, that sample preparation and quality check is very important. We have to make sure that peptides are really well digested. Peptides are being taken forward, and those are quite clean. If we fail on this step, all the previous experiments and steps will be of no use, and subsequently mass spec cannot give you good data set, and that we do see a lot of failure on this very crucial juncture. It's very important to learn the peptide cleanup, the removing the salt, and making sure that from the C18 material peptides are bound. Then we elute them out, and then only we can take those for the further mass spec analysis. Prior to doing that, we again quantify the peptides. This thing again we'll talk in one of the classes, when we can show you the peptide cleanup and peptide quantification, which can be injected to the mass spectrometer. These will be very small steps. So this gives you the idea about the proteomic sample preparation, and also some idea for what is we are going to do in the project about the data analysis part. So that's pretty much from my side right now. Let's take a couple of questions or comments if you have any. And in the meantime, TS get ready to share you the file which we would like to analyze by all of you. So any questions, any comments from the sample prep part, or from the presentation and the project which we have just explained you? Students, please ask queries now. Don't send a lot of emails afterwards. It's a good idea to discuss these things in the interactive class. Ashi, are you clear about what we spoke for the project and presentations?
Yes, sir. More or less, I understood everything.
Okay, all right. What about Devanshi and Dhanush?
Yes, sir.
Yes, sir.
All right, we are clear about it. So good. Looks like my presentation was very clear then. Okay, Jyoti and Malo, you have any questions from the sample prep?
No, sir.
We are able to follow up the different ways of sample preparation methods and quality control checks?
Yes, sir.
Okay, all right, good. Shashwati, in the meantime, have we shared the file?
Yes, sir. I have shared the file in the chat.
Okay, and you need to also show the time on the screen. Maybe reduce the time a little bit. Maybe give 8 minutes or so. Yes, Aparna, please ask question if you have any.
Yes, I wanted to know whether there's any specific reason to use in-gel or gel-free sample extraction? Like what are the how do we select which technique to go by?
No, I think very good question. So preferred way will be in solution, because if you have a complex proteome and from that complex proteome sample you want to identify all the protein, so preferred way will be in solution digestion, because that is less cumbersome and having less step. Otherwise, if you have to run the same complex proteome on a gel piece, you have to make at least 10 or 15 pieces to cut the whole gel into small pieces and then do each of those separately in gel digestion. Then only you can put that together to get the complex proteome from one lane of SDS page. Whereas in the in solution, it's simply just you take few microliter of your protein extract and do the steps, and that will be reducing a lot of your work. And of course, when you do the 10 different times sample processing, versus when you are doing it once, you are going to lose less peptides. So therefore, in solution is more efficient and more effective for the complex proteome analysis. Many times people are only interested in a specific band of interest or a spot of interest or a specific region of the proteins. Then you are better off doing the in-gel digestion. Also, sometime due to some regions, your protein extracts can only be in a specific type of buffer, which may not be completely compatible. Like you have too much high SDS in your sample prep. Like if we are trying something on FFP, for example, and it requires a lot of SDS. So then if too much detergent is there, that is not going to be compatible for the LCMS analysis. So again, you can separate them on the SDS page gel. And after running from the gel, then all those interference has gone. And then you can clean up these gel pieces and you can proceed for the in-gel digestion. I hope that gives you a balanced perspective of choosing both. Like in-gel can be taken for the extreme type of buffer condition from the complex proteome samples, or to look at the specific proteins of interest or spots of interest from the 2D gel. Whereas the in solution could be generic thing which can be tried for any complex sample. And if your procedure works well, it's very less cumbersome, more efficient, and you can get to see thousands of protein from that digest. So I hope that answers your question, Aparna, or anything else you want to clarify?
Yes, sir, that clarifies it out. Thank you.
All right, sounds good. So let's start the data file which has been given to all of you. Please start analysis with the MetaboAnalyst, and time given is 8 minutes to you. Go through the steps the way we have done in the previous steps, and then we will talk about your output and what is expected from that analysis. Yeah. So your time starts now.
Hello, sir. Can you
Yes.
please send the file again?
I think
Yeah, in the chat. I can't see it in the chat.
Shashwati, please redo that.
Yeah, I have shared once again the test file. You can download and try.
Okay, so the time is up. So if you have faced any issues, then you can ask me. Otherwise, I'll walk you through the entire process once again.
Yeah, Shashwati, you can share your screen and just quickly walk through the steps again. And if they have faced any problem, then they can clarify.
Yes, sir. Yeah, so I hope my screen is visible. So this is the MetaboAnalyst website. So you have to click here to start. Then we'll go for statistical analysis to get our significant proteins. So here you have to select peak intensity. Then samples were in column unpaired. So I'll choose the test data set and click submit. It will take some time. Yeah, so here you have options. So you can proceed without missing value or with missing value. So we'll do the missing value imputation here. So first we have to remove the feature which has greater than 30% of missing values. Then we have to do the missing value imputation via KNN feature wise. Then we'll click on process. Yeah, so we'll skip the data filtering part here. We'll simply click process. Then in the sample normalization, you have to first do the log transformation, and you can opt for any other kind of normalization. For now, I'll do the median normalization. You can click on normalize. Okay, so in view result, you can see how your feature-wise normalization look like and how the samples were normalized. Okay, so all the samples are well normalized here. Then we'll click on process. Yeah, so here you have many options. So first, what you can do, you can go for fold change analysis here. So here I have kept two, and you can choose tumor versus healthy. Click submit. So now you can see the down-regulated and up-regulated protein, which are a fold chain more than 2. So this blue are the down-regulated protein and red are showing the up-regulated protein. Then you can do the t-test analysis. Here you have to choose p-value 0.05. You can do the raw or FDR adjusted p-value. For now, I'll do the raw. Group variance is equal. Yeah. So these are the protein, like 480 of the protein which are passing the p-value criteria here. Then we can go for the volcano plot analysis, where you can do the both, like the FC fold change and the p-value. So here I'll set the fold change as 2. Then I'll choose tumor versus healthy, and in the p-value I'll type 0.05. Raw and group variance is equal. So I'll click on submit. Yeah. So here I got 187 down-regulated protein and 223 up-regulated protein from this volcano plot. So you can go to the table, and from here you can actually download the result file for the volcano, right? Also, if you want to see any protein which are upregulated or downregulated of your interest, you can just click on view here. So then in top, it will show the expression, like the box plot of this protein. Now you can see healthy versus tumor. The tumor, this protein is downregulated for the tumor. Yeah, so then you can go to the PC plot. Here you have to click on 2D score plot. Yeah, so here you can see two groups, healthy and controls, are showing two clusters here. Then you can go for dendrogram. So here also you can see all the tumor and the healthy samples are all together, right? They're not like tumor samples and healthy, all are well segregated. Then you can go for the heat map also. So here in the heat map, you can use the top 25 protein, which are segregating your two cohorts. Then you will got heat map for top 25 protein, which are segregating your whole data set, right? Okay. So in the download section, you can go for whatever you did, you can download all the files at a time. So here all the files are available. So if you want to do the reactome analysis, you can download the Volcano table. So, okay, I'll download the Volcano table. I'll just open. Yeah. So here you have all the list of protein which are up or down regulated that are significantly altered in our list. So you can just take this list of protein for your reactome analysis. And this is the FC and log2 values all are here. So now you can proceed for the reactome analysis. Then we'll discuss about the reactome analysis.
Excuse me, Shashwat.
Yeah.
While we were doing the the exercise, we had taken the group variance for t-test to be unequal. So how do we decide that we have to take the group variance to be equal or unequal?
Yeah, so here in the test data set, you have eight samples, right? So 8 tumor and 8 healthy samples. So for that, you have to select equal. If you have like odd number, like 8 or 7, then you can choose unequal.
Okay, thank you.
Yeah. Okay, now I'll give 5 minutes for the reactome analysis. You can go for reactome, then we'll discuss. Okay, so the time is up. I'll show how to do the Reactome analysis. I'll walk. Just a second. Okay, so for Reactome analysis, we can go for the analysis tool in the Reactome website. It will take a time. Yeah, so here there is different types of options. So first is analyze gene list or analyze gene expression. So first I'll show the gene list, like the protein list that we got from the Volcano. Yeah, so these are my all protein list. I'll just copy. Continue. So here we can include the interactors also. Click on analyze. Yeah. So here on the basis of p-value, I'm getting all the pathways. So we can go to the download. And here we can download the PDF version of the analysis. Or else we can here also download the .csv pathway analysis result. Okay, first I'll go for the PDF report. Okay, so take some time. Yeah. So in this report, we'll go to the most significant pathway. So here you can see like first is neutrophil degranulation, platelet degranulation. These top pathways are coming. So what you can do, you can go to this particular pathway by clicking here. Let's say neutrophil degranulation. So here it will show the description about that pathway. Then here you can see the entities found from this pathway, right? So there are as many entities from our dataset which are mapping to this pathway. So from this you can select some of the protein, or else what you can do, you can just select the top upregulated proteins from your Volcano. Like I'll select from here. Yeah. So here this is regulation. Yeah, so this protein you can see this is the top upregulated protein, that some of the top upregulated protein you can then take and you can check in the next plot how this protein and for the unique peptide of this protein. Or else what you can do, just a second. Yeah. From the result file that you got from the Reactome, here also you can check for the top pathway and how many entities are found from that, and all the protein that are mapping to that pathway. You can take those proteins, some of the top upregulated or downregulated protein from those pathway, and you can check in the next part also. So that you can do next. That will be your targeted approach. Or you can take those protein for your skyrine analysis, and we can target those proteins, so that we'll see this protein are upregulated or downregulated in our samples or not. Another way could be you can go for Gsa analysis. So in the Reactome, here is the analysis Gsa expression. So here you can choose the Gsa one or camera one. Here you can add the data set. So here you have to add the intensity data set. So from the MetaboAnalyst, you can download the data normalized file. So that normalized file itself you can use. So I have the normalized file, test normalized. So you can upload that file and you can annotate here in the Reactome. Let's say it's my healthy sample. You can annotate all the healthy sample as healthy. And all the tumor samples as tumor. Yeah, so we'll click on continue. Then we'll select healthy versus tumor comparison, right? So the data set is added now. We'll click on continue, continue. It will take some time and then it will give your top upregulated pathways and the downregulated pathways from your entire data set, right? So it will take some time. So you can go to the PDF of this analysis. So here you can get the information about the top upregulated pathways like gluconeogenesis. So it is the downregulated pathway. It's showing the downregulation. Likewise, you can go and you can select the top protein which are falling in those pathway and you can go for the targeted analysis. Okay, now here what you can do, you can go to your Volcano Excel sheet and find the top upregulated protein and check the digestion for that protein in the next plot. Okay, I will give you another, you can do it within 3 to 4 minutes. I will give you 3 minutes, then we'll discuss about the next plot, right? Take the top upregulated protein from your Volcano for now time. Okay, so the time is up. So I'll show the next part analysis. Just give me. Yeah, so we can go in the next plot. Here, in the next plot, you can go to a tool. So here you can do many things. So for now, we'll do the protein digestion for our top upregulated protein. Okay, so from the Volcano, this is my top upregulated protein. Yeah, I'll click on digest. So it will show impossible to get the data, but it will give us the data. So do not worry for that. So it will take some time. Okay, so it will take a little bit of time. Yeah, so here it will give a different option for protease, different protease options. So mostly we do the digestion using trypsin. So you can click on trypsin, and from that you can click on get peptides. So here you are getting total 47 entries for this protein, right? So 47 peptides are coming. So here you can check unique without variant or not. It's showing here. So you can check which peptides are unique from this, and you can take those protein forward for your Gsa analysis. And I'll show the Gsa analysis. So just a second. If you have already installed the Skyline Daily, you can try out this also. Yeah, you can go to Skyline, and setting we have already shown you in previous classes, all the settings. So you have to set the background proteome as human proteome and all the settings that they have already shown to you. So the minimum peptide length you can keep 8, or maximum you can keep 20, like that. Okay, so you can go to insert protein name. So I type the protein name here. Yeah. So here you can see it is giving me all the description, the name of the protein, what is the protein, ATP dependent RNA helicase protein, and its sequence, its accession ID, gene, and from which species it's coming. So you can click insert. So it will give all the peptides for that protein. And here you can see all the peptide precursors and total transition list. And you can export this transition list, and you can analyze or this protein, and you can know if this particular protein is actually upregulated or downregulated in your tumor or healthy samples. So that's it. That's it from my side. So
all right, so now I think Deep could show the project 2 possibility, the computational project, quickly.
Hello.
Yes, Deep, go ahead.
Yeah, I'm sharing my screen.
This is for the computational project, though the student who would like to opt more from the, not the biology, but more computational project, they can watch this one more carefully.
So the project which we have given a broad title, which is which is computation or in silico transcriptomics data analysis for identifying the differentially expressed genes. So this project is under the Human Brain Disease Network. So I think I have given you guys some idea regarding the Brain Prot, which is a standalone portal that we have made from our lab in collaboration with Spain, where we have ended that particular portal with a message that even the important genes and proteins which we know that are markers for different diseases has some differential expression or have some basal level differences in terms of different regions of human brain. So from this particular background, we got some idea that even we can try to see how the the human disease network in terms of brain diseases are actually linked with. So we got some idea from one of the manuscripts that were published in PNAs, where they have tried to jot down the human disease network, and with they have tried to get the name of the genes. So as you can see, disease gene network. So where they have got the name of the diseases and the gene that are involved over here, and they have tried to make a mega network of the different disease phenome and disease genome. So this is something which gives us an idea that the marker genes which is present in different diseases, how much popular those marker genes are in other diseases, whether there is a correlation between one gene with the other diseases or not. So taking this thing forward and the idea and also the foundation that we have got for the brain protein project, we have did some text mining analysis for understanding the relation between the human brain diseases. And for example, PD is Parkinson, Alzheimer's disease, multiple sclerosis. So these green colored diseases are mostly the neurodegenerative diseases. However, glioma, astrocytoma, medulloblastoma, meningioma are brain tumors. So as we know that the brain diseases are mostly differentiated in two types. One is neurodegenerative disease, another is the brain tumors. So we are trying to understand using a text mining procedure of the gene as an input query, that what is the correlation between the neurodegenerative diseases and the brain tumor, and also how much close one disease is with the other disease. So we got a very good idea from this, and from this one only we have taken this thing forward to understand that whether we can mine the different kind of transcriptomics or gene expression data from GEO, gene expression omnibus. For example, you guys know about proteome exchange, which gives you an accessibility of the proteomics data. So for example, the same way gene expression omnibus and array express gives you a list of gene expression data or transcriptomics data that which already people have published. So the goal is to download some of the data and reanalyze those data and try to understand the correlation between data set to data set. So this is something global metadata analysis in a broad spectrum in different diseases. So we have already did the data mining. So as you can see how we have make a sheets like this where each data sets are there, what kind of omics analysis has been done, what platform is there, how many samples are there. So all these things are already done. So if some of you are interested, so this project will start from here. So what you guys have to do, so we will give you a GAC ID. So this particular GAC ID will have the the list of all the samples, raw files of the transcriptomics data. So you will go GEO, gene expression omnibus. You will search this GAC 43290, and once you have searched this thing, you will get the raw .tar file. Like that means this particular data set and all their raw. So you need to download this raw files. Once you have downloaded, it will download in a zip version. You will unzip this particular raw files. Once you have unzipped, you will get each raw file in a zipped version. For example, like this. So once you will open one zip file, you will get one single .cl file, which is the raw files. So once this is done, you will unzip all the file and you will make a folder of all .cl file. So the first step you will do is the downloading the raw files and unzip files into a folder. So as you can understand, this is not a big thing. It's a very first step of the analysis that you need to do. That means you are taking all the raw file, accessing all the raw files. So once this is done, the next part is that you know about the raw files, but you don't know that these raw files belongs to which grades or which groups of the sample. So what we will do in the sample annotation and for the grouping in the GAC only, when you will select this particular data, you will get a tab over here, analyze with GO2. So once you will get these things, you need to click over here. So it will give you a big sheet which we call as meta file, sample meta file. That means this particular accession ID. So these accession ID are nothing but the raw file ID. So as you can see, GSM 1060065. So GSM 1106065, right? So these are nothing but the raw files. So that means each raw file's information are given over here. For example, these raw files belong to meningioma tumor. The gender is female. The age is 62. The cytogenic group deployed. Grade is 2. Histology is atypical. So like this, all the informations are available over here. So from here, now we need to select only that information that we need. For example, in most of the cases, we will do grade 2 versus grade 1, low grade versus high grade, something like that. So that means you need to select this one and as a Excel sheet. So that means we need to prepare a matrix file like this. But these are the accession number and this is the WHO grade. So this is important because this will require in the further analysis. So what we did first, we have downloaded all the raw files and unzip the raw files, and then the next part what we did is that we have taken the annotation grouping or the sample grouping out, and we have prepared a matrix file. So now the next third step is that we need to check the quality of the data. That this is very important because someone has uploaded the data. We don't know how the quality looks like. So what we will do, we will go to an array quality matrix. So this is something which requires a little bit of our hands-on. It is very easy, nothing like that, because the codes are already available and everything is over there. So it is presented in the bioconductor, and even over there also, different our documents are available. So first we will see sample to sample expression, then whether there is an outlier or not, and once the analysis is over, it will give you a complete complete file, which will give that which files we need to which are acting as an outlier. So for example, this 1, 2, 3, 4, 5, 6, these 6 are nothing but 6 different labels of analysis that they have done, where they have tried to understand that which are outliers. So just let me go and show you. So for example, this is the data set, and this if I am clicking over here, so it is giving all the files, raw files that you have uploaded, and how these raw files in 6 different level of quality check are whether they are detecting as an outlier or not. So 6 different step means, so number 1 is the correlation between the array comparison. So as you can see, in between this 19 to 19, there's a star is there. That means this is the outlier is coming. So if I go to the next one, maybe 2. So 2 is something which is a box plot of the normalization it has shown. If I am clicking in this figure, so these are the samples which are there. And if I come down over here in the outlier, just a minute. So the third one. So yeah, so yeah, this one. Yeah, so see, there is 2 stars are there. So this means these 2 data are outlier data set. So like this, we will see this particular this particular table, and we will remove these samples, right? These samples which are coming as an outlier. So this is very important, because on the basis of that only we can take the data forward. So after removing the data, we will again put this particular file, the raw files, into the array quality matrix, and we will cross check whether the data has any other outliers are showing or not. So generally it don't show. So this is once this particular outlier detection is done and you have removed the data. So the next part is coming the normalization, data normalization, which generally we use the RMA, which is a robust multi-chip average normalization for for your microarray, which is comes under Lima package. So this particular RMA normalization is mostly available in the this particular the next step which I will show, which is the FELM GUI. So what it actually does is that once the normalization is done, once the outlier detection is done, you will put the data over here and it will do the normalization. And it's a good practice that once the normalization is also done, this particular normalized data can be taken forward, and we can also see in the array quality matrix that whether after normalization is there any outlier is detected or not. This is also important. But once data normalization is done, then we will move ahead with the statistical data analysis in the FELM GUI. So once you are, you will write some initial code, it will give you a GUI, that means graphics user interface. And over there, as you can see over here, the available RMA is already there, right, for the normalization. So you will put the data, and finally the data will give you, these are the probe ID, the symbol, top differentially expressed gene, right? This is the name, then this is the p value, their fold change will be there, Bonferroni correction will be there, all this information it will give. So if I just go back and see, there is only 2-3 steps are there. For example, this one step is there, and this is another step is there, where little bit R is required, right? Like the R and R Studio is required, where you need to put some little bit of some codes, just the change of the name of the files, and that's it. And the analysis will be done. So once this part is over, you will get the differentially expressed gene. This is nothing but same as the differentially expressed protein. So the next step is same, where you will take the P cross N matrix of the normalized data, and for example, mostly in proteomics data, you were choosing this one, right, where the intensity based on quantitative proteomics data you were choosing. But for this one, we will choose the microarray normalized data, where the normalized and log2 transform microarray based gene expression values are there. So this is the a simple workflow which I showed, where you can take the data out and you can go ahead with the analysis to get some more idea, and that we can take forward and try to correlate with the diseases, different kinds of diseases, and you all will get some different GAC ID of different diseases, and which you will go ahead, right? So if anybody will be like interested, then we'll give some more demo regarding the workflow in the next classes. Thank you.
All right, so thank you, Deep. In fact, students who are interested in this kind of project, they can go through it, and we will have more, clarification in the subsequent class. Right now, what we are going to do, we are going to share you a Google form. Please fill the form stating that which of the two type of project which we have explained to you, you would like to prefer to do that for your subsequent project activity and presentation. Because we need this clear answer from each student. Accordingly, we have to prepare the number of topics and stuff like that. So Shashwati, please share the link now, and student, you can fill the form and then you can leave. So thank you very much. I hope you are getting excited that you're going to learn a lot of new things independently now, and we'll be also keen to see like how your learning can actually result into some interesting presentation and ideas which we can discuss after the midsem. That's all for the day. We'll close it. Please finish the form, and we will share you over the weekend another form which will have more project details. All right, that's all. Thank you.